Welcome to today's MedPhysio tutorial on neutrophil function, often called the body's rapid response team. When an infection breaks out, neutrophils are the first immune cells to arrive. They're powerful, fast, and short-lived. At the end of this session, you'll not only understand what they do, but also how they do it with remarkable precision. In this tutorial, our goal is to walk through the entire life cycle of neutrophil. From the moment it senses distress, and the moment it helps resolve inflammation. You learn how neutrophils migrate towards infection, how they bind and engulf microbes, and how they deploy powerful oxidative and enzymatic systems to kill pathogens. We also explore NET formation and how programmed neutrophil death promotes resolution. Finally, we'll connect these mechanisms to clinical conditions such as neutropenia and chronic granulomatous disease. Now, by the end of this session, you should be able to map on the entire neutrophil response. It's a seamless physiological storyline from chemotactic signaling to microbial destruction, final resolution. You will be able to explain why each step occurs, not just what happens. Also be able to describe oxidative versus non-oxidative killing with precision, including the roles of NADPH oxidase and myeloperoxidase. Moreover, you'll understand mitosis and its double-edged clinical significance. Most importantly, you'll be equipped to interpret clinical patterns such as leukocytosis with left shift, recurrent infections in CJD, or impaired killing in MPU deficiency, and link each presentation back to the underlying neutrophil mechanism. Neutrophils are the frontline warriors of the innate immune system, highly mobile, short-lived, and relentlessly effective. They constitute about 60 to 70% of circulating leukocytes, forming a vast, ever-ready reserve that can be rapidly mobilized from the bone marrow storage pool when infection or tissue injury occurs. Physiologically, their presence signals the immediate, non-specific phase of defense. That is a phase that operates before antibodies or even memory cells are even activated. Within minutes of an inflammatory trigger, chemical messengers such as interleukin-8 and leukotriene B4 establish a chemotactic gradient that calls neutrophils to the affected site. Though they survive for only 6 to 12 hours in circulation, each cell undergoes a precisely programmed sequence of functional transitions: chemotaxis, diapedesis, opsonization, phagocytosis, killing, and resolution. Think of this as the CDOPKR pathway. In essence, the neutrophil represents speed and sacrifice. A cell that is designed to act first, fight hard, and die young, ensuring that the more specialized arms of immunity have time to prepare their response. Now, when explaining this in exams, always tie structure to function. The multi-lobed nucleus allows for extreme deformability for migration. The cytoplasmic granules serve as biochemical arsenal for microbial killing, while the short lifespan limits collateral tissue damage. Together, these make the neutrophil an example of how form and function align perfectly in defense. Chemotaxis is the process that guides neutrophils toward an infection or injury. When tissues are damaged or invaded by microbes, they release chemical signals and also cytokines like interleukin-8, complement fragments such as complement factor 5A, and bacterial peptides. These chemoattractants create a gradient pointing to the exact site of trouble. Neutrophils detect these signals through surface receptors and sense which direction the concentration of chemoattractants is increasing. They then rearrange their cytoskeleton and crawl towards the strongest parts of the gradient. This guided movement ensures neutrophils reach the pathogen quickly and prepares them for adhesion and diapedesis. Now, for exam insights, when describing chemotaxis, emphasize the stimulus and the response link. Look at this path. Injury leads to release of chemoattractants, leads to gradient formation, leads to receptor activation, and then this cytoskeletal movement and directed migration. So after chemotaxis brings neutrophils close to the inflamed region, the next challenge is crossing the vascular wall, which is called diapedesis or transendothelial migration. When tissues release inflammatory mediators like interleukin 1 and TNF alpha, the nearby endothelial cells become activated. This activation makes them express selectins and intercellular adhesion molecules, that's ICAM1, on their luminal surfaces. Now, this is the neutrophil. You can see it has the sialyl Lewis X modified glycoprotein and integrin on its membrane. Now, as the neutrophils circulate, they begin to roll along the endothelium, transiently attaching and detaching through selectin sialyl Lewis X interactions. You can see that here. This rolling slows them down, allowing integrins on the neutrophil to bind more firmly to the ICAM1 on the endothelial cell. That happens here, integrins and ICAM1 binding more firmly on the endothelial cell. Now, once firmly adherent, the cell flattens, reorganizes its cytoskeleton, and begins to squeeze through endothelial junctions. This is this transmigration step. At this point, a molecule called PECAM1, that is platelet endothelial cell adhesion molecule, found at the endothelial junction on the and on neutrophil membranes, acts as a molecular compass, guiding the cell through the narrow intracellular gaps while preserving the vessel integrity. The entire process is energy dependent, involving calcium signaling and actin remodeling, ensuring that even though thousands of neutrophils may exit, the vascular barrier remains intact. So, neutrophil attaching selectin, sialyl Lewis X glycoprotein, rolling, integrin, ICAM1, binding, reorganization of the shape, and PCAM1 and diapedesis. An exam tip: use the triad rolling, adhesion, transmigration, and mention the key molecules at each stage. A well-phrased answer will begin like this. Now, following chemotactic activation, neutrophils adhere to cytokine-activated endothelium via selectins and integrins, then transmigrate through become one mediated junction into tissues. When neutrophils reach area of infection, we must first identify which particles are harmful. This is made possible through opsonization, a process that prepares microbes for ingestion by coating them with special molecular tags. These tags, known as opsonins, act like flags that signal to neutrophils that the particle is dangerous and should be engulfed. The most important opsonins are the immunoglobulin G antibodies, especially the G1 and the IgG3. They complement fragment C3b and C-reactive protein, all of which bind firmly to the surface of microbes. Neutrophils carry receptors that specifically recognize these opsonins. FCY receptors attach to immunoglobulin G, while the CR1 receptors bind to C3b. When these receptors engage with their matching opsonins, the neutrophil forms a strong, stable connection with the pathogen. This binding triggers internal signals that activate the cell and prepare it for phagocytosis. By transforming random encounters into precise, high-affinity recognition, opsonization greatly increases the speed and effectiveness of microbial clearance. Clinically, this system is essential. If opsonins or their receptors are defective, as seen in complement deficiencies or FC receptor disorders, neutrophils may still arrive at the infection site but fail to recognize or attach to bacteria, leading to recurrent or severe infections. The next stage is phagocytosis, which begins with recognition and engulfment of an opsonized microbe forming a phagosome. The phagosome then fuses with lysosomal granules, forming a phagolysosome, where killing occurs. Now, within this chamber, two powerful systems come into play. One, the oxygen-dependent killing mechanism called respiratory burst. Oxygen-dependent killing uses NADPH oxidase and myeloperoxidase to generate potent oxidants like hydrochlorous acid. The second is the oxygen-independent killing, which relies on enzymes such as defensins, lysozyme, and lactoferrin to degrade microbial structures. Defects in these mechanisms, especially the nicotinamide adenine dinucleotide phosphate oxidase, that's NADPH oxidase deficiency seen in chronic granulomatous disease, leads to recurrent and severe infections. Now, when explaining this in exams, it helps to write it as a logical chain. Following opsonization, neutrophils engulf microbes into phagosomes, fuse them with lysosomes, and destroy the contents through oxygen-dependent and oxygen-independent mechanisms mediated by NADPH oxidase and myeloperoxidase. Simple. Neutrophil extracellular traps, nets, are released during netosis, the unique death process where a neutrophil expels a web of chromatin and antimicrobial proteins. Strong stimuli such as bacteria, fungi, interleukin 8, TNF alpha, or activated platelets trigger nuclear membrane breakdown and chromatin decondensation. The DNA mesh, coated with histones, myeloperoxidase, elastase, and other enzymes, forms sticky networks to trap and immobilize microbes. This allows neutrophils to keep defending even after death by creating a localized antimicrobial barrier. However, excessive nets can damage tissues, contributing to conditions like systemic lupus erythematosus, vasculitis, and sepsis. Exam tips. Now, when asked about nets, structure your answer around three key points. One, origin, from activated or dying neutrophils, netosis. Two, function, extracellular trapping and killing of pathogens. Three, pathology, excessive nets leads to inflammatory and autoimmune tissue damage. Once a neutrophil has completed its mission of engulfing microbes, using enzymes or even forming nets, it undergoes programmed cell death. Unlike necrosis, apoptosis preserves the integrity of the surrounding tissue. Cell membrane remains intact, its contents are neatly packaged, and there is no leakage of damaging enzymes. These apoptotic neutrophils then send subtle signals that attract macrophages. Macrophages engulf and digest them in a process called phagocytosis. Phagocytosis is a biological peace signal. When macrophages ingest apoptotic neutrophils, they switch their own behavior from pro-inflammatory to anti-inflammatory. They release mediators such as transforming growth factor beta, that's TGF beta, and interleukin 10, which suppress further neutrophil recruitment and stimulate fibroblast activity, angiogenesis, and tissue repair. Thus, the resolution of inflammation is an actively regulated process orchestrated by dying neutrophils themselves. For exam tip, when describing resolution, link the cell fate to function. An ideal phrase might be following pathogen clearance, neutrophils undergo apoptosis and are removed by macrophages through phagocytosis, leading to the release of anti-inflammatory mediators and the initiation of tissue repair. Failure of this resolution phase underlies chronic inflammation, seen in diseases like rheumatoid arthritis or COPD, that's chronic obstructive pulmonary disease, where ptotic neutrophils are not efficiently cleared. For memory tag, you can use arrival, attack, apoptosis, the three A's of neutrophil life. Our clinical correlates: when neutrophil numbers fall, as in neutropenia, body loses its first line of defense. This may occur after chemotherapy, bone marrow suppression, or severe infections. Clinically, patients present with recurrent bacterial and fungal infections, mouth ulcers, and poor wound healing. In such cases, remember, it's not the microbe that's changed, it's the missing neutrophil army that tips the balance. Conversely, leukocytosis, an increased neutrophil count, is a hallmark of acute bacterial infection or inflammation. Cytokines like gene, granulocyte CSF, and interleukin 1 stimulate bone marrow release of stored neutrophils, even pushing immature forms into circulation, which is a finding often referred to as a left shift. In exams, this is an easy mark. Cytosis with left shift suggests acute bacterial infection, remember. Now, the quality of neutrophil function is just as important as the quantity. In chronic granulomatous disease, CGD, a defect in the NADPH oxidase enzyme cripples the respiratory burst. Neutrophils reach the site of infection but cannot generate reactive oxygen species, so microbes survive inside them. The immune system compensates by walling off the infection, forming granulomas, hence the name. Similarly, myeloperoxidase (MPO) deficiency impairs conversion of hydrogen peroxide to hypochlorous acid, inducing oxidative killing. Most patients remain asymptomatic, but severe cases may develop persistent candida infections. Subtle yet classic exam link. Now, as your exam memory tag, numbers vs function. Too few cells, neutropenia, leads to infection risk. Too many cells, that's leukocytosis, leads to shows acute inflammation. Defective oxidative burst can lead to CGD, while defective MPO leads to reduced but partial killing ability. Let's bring everything together. From chemotaxis, where it senses distress and races towards danger. Diapedesis, where it skillfully crosses the vessel wall. Then opsonization and phagocytosis, where recognition meets action. And finally, killing and resolution, where it ends the battle and restores peace. Each step is distinct, related, and essential. These 6 pillars, C, P, O, P, K, R, form the core memory map for neutrophil physiology. Supporting them are net formation and the extended reach of dying neutrophils and apoptosis, which ensures inflammation concludes safely. Together, they show an organized balance and remind us that immunity is not just about destruction. Now, clinically, each phase leaves diagnostic clues. Low numbers, as in neutropenia, um, predisposed to infection risk. High counts, leukocytosis, um, shows an acute bacterial response. Effective NADPH oxidase can lead to CGD, and faulty myeloperoxidase leads to incomplete killing by the neutrophils. Now, a mini activity which can serve as a self-check review, as the questions listed here. Take 2 to 3 minutes to recall these answers without notes, because active recall is how memory is strengthened. Thanks for listening. See you in our next video.